we get to making glycolysis. And it kind of has to be that way, because if you're going to make gluconeogenesis irreversible, so that when you're going backward, going up the pathway, it's actually thermodynamically strongly favored to go up. That's a direction that is unfavored thermodynamically. So the only way to make that work strongly in that unfavored direction is to use a lot of ATP. Right? So that's what we have to do. And you can see the beginning of that right here in the sense that, per glucose, you made 2 ATP here, which we just used up 4 of them here. And all we did was get back 1 step. Okay. Question? Yeah. I'm sorry. Yeah.
Why is GTP used instead of more ATP in the second step of?
I have no idea. I mean, it's
And another question is, what is the transport mechanism that moves pyruvate into mitochondria?
The mechanism is through a specific transporter protein that there's a binding site for pyruvate on the outside, and then there's a conformational change that opens up a pore that's specific to pyruvate. Usually those processes require some ATP to drive the conformational changes, not always. But yeah, it's something like that. Yeah. Yeah, so why GTP is because some enzymes differentiate to use GTP. I don't know why this particular one is that way, but as we discussed last time, when we say GTP, we know that there's always enzymes around to interconvert them, GTP and ATP, for example. And those kinds of enzymes are typically in both compartments, cytosol and mitochondria.
So with the transportation, does it use ATP then to also transport it back out of the mitochondria?
Well, I shouldn't say it needs it for the transport process. The book does not talk about that. I'm just kind of saying in general, sometimes the transport of metabolites through membranes is a process that requires ATP because the receptor has to change structure so much. We don't have any discussion of that in the text for these, which probably means that in these cases that we don't need to use ATP.
Okay.
Yeah. Thank you. So it's safer to just, let's just say that they don't need ATP. But it is a specific binding process and some kind of structural rearrangement. So the binding energy by itself would be enough to drive the rearrangement of the structure to get the pyruvate to move through the bilayer. And there would be some kind of channel. I don't know if you guys must have studied this a little bit last time with membrane proteins. Channels are opened up so that molecules can go in and out. Did you talk about ion transporters or anything like that? Yeah, so one of those processes. Other questions? Okay, so here's the cytosol and here's the mitochondria sort of depicted. I don't know how well you guys can see it. I can't see it too well because I'm off to the side, but you guys can't see it well either. Anyway, there's a boundary here for the mitochondria, and this is cytosol up here as well. So pyruvate can get in. Pyruvate gets into the mitochondria. If it's in the mitochondria, as you know, what we know so far happens to it is the pyruvate dehydrogenase complex grabs this guy, right, and it doesn't add CO2 to it, it takes CO2 off of it, right? So, what's going to stop the pyruvate dehydrogenase complex from grabbing this pyruvate? It's going to have to be regulated, right? It's going to have to actually be off. And why would it be off? Well, because there's probably already a lot of energy in the cell. And how do the relevant proteins know that? Because there's a lot of ATP around. Or a lot of molecules that are made from ATP or that also signal a high energy state in the cell because of other metabolic processes. Does that make sense? So it's not shown here, but we know this pyruvate is supposed to go to acetyl-CoA, right? It's supposed to go. Because what we've learned so far is we're breaking stuff down and we're using the energy of glucose. But now we're not using the energy of glucose anymore, right? We're saying we're already in the high energy state. We don't need the energy from this fuel molecule. We're going to make more fuel molecules. And we're not even going to make the fuel molecules themselves unless we're the liver, in which case we're going to make them and export them. We're going to make the fuel molecules and then we're going to store them as glycogen. So gluconeogenesis goes back to glucose, or a related compound to glucose, and then from there, there's another pathway to make, much shorter pathway to actually make the glycogen. Okay, so here we are going up. So PDC is turned off in ways we'll talk about later. So there's a carboxylase enzyme step here that goes from a 3-carbon compound to a 4-carbon compound, oxaloacetate. And this is pyruvate with a CO2 on it. So if you look at the structure of oxaloacetate, you will see that it's pyruvate with a CO2 on it, or a carboxylic acid on it. But if you add CO2 and form a carbon-carbon bond, then you get to make it, you make oxaloacetate from pyruvate. So that's what this enzyme is doing, it's adding. I mean, it wouldn't be binding gaseous CO2. It says CO2, but in that aqueous compartment it would be carbonate, and it would work through carbonate. Oxaloacetate then goes to malate inside the mitochondria still. Here's the boundary. And so this is malate dehydrogenase, which is the same enzyme that's in the TCA cycle. The only difference is it's going the other way. And so if you go back to your TCA cycle and look, you will see that malate to oxaloacetate, right up near the end of the cycle, in fact, step 8 of the cycle, the last one, is reversible. Right? So if we're going to do gluconeogenesis, we are using the capacity of the malate dehydrogenase to run backwards, to make malate from oxaloacetate. And what that does is uses up an NADH, because now we're reducing the oxaloacetate to malate, right? So if we reduce it, right, we're in a sufficiently energy-rich state that we don't need this NADH to go off and make ATPs on the intermitochondrial membrane anymore. We're happy to use it up in order to reduce a metabolite in a redox reaction on an enzyme so that we can make the right metabolite, which is then transported in another specific transporter in the membrane that goes out to take the malate out. And then in the cytosol, there's the same enzyme, right? It's an isozyme, it's got some sequence differences, but in the cytosol it's running the other way. It's running exactly the way it does in the TCA cycle, right, to generate NADH in the cytosol. And we need to regenerate NADH in the cytosol, right? If we're coming from lactate, we can regenerate NADH here, right, in which case the pathway is different because there's some signaling back and forth that the cell will know, so to speak, whether it needs to go this way or this way. But we know we need to regenerate NADH to go this way, because if we don't, we can't run the glyceraldehyde 3-phosphate dehydrogenase reaction in glycolysis backwards, right? That's step 6 of glycolysis that made NADH. Well, now we got to make NAD. Right? Yeah.
I have a question about the transport of malate from the book saying it's in the inner mitochondrial membrane.
Yeah.
So how does it also get through the outer membrane?
Yeah, there's no selectivity in the outer membrane. So the outer membrane is pretty porous to all these metabolites. So as far as we're concerned, which is largely the case, there's no selectivity in getting in and out of here. So it's not a completely permeable compartment in the sense that everything in the inner membrane immediately goes, anything in the perimetotic space immediately goes out, but it's not selective either. So if we're driving a process whereby malate comes through the inner membrane, it readily diffuses out of the outer membrane as well. Okay, so malate to oxaloacetate, and then another enzyme here, the carboxykinase. So the two enzymes that are unique, that are shown on this slide here, carboxylase and carboxykinase. And you notice here it says mitochondria always. This one could be a cytosolic or a mitochondrial enzyme. Those two enzymes here are here at the beginning and here at the end. And that previous slide didn't mention these two. It didn't mention them because they're not new. So they're sort of, the authors of your book are not giving you the whole thing right away. You're saying, here are the new ones, these two, right, this and this. But malate dehydrogenase, you already know about that.